three approaches can give a significant gain in the accuracy if I use multi-breed selection scenario, like with the shared SNP effects. And the thing was, even in the real data, the accuracy, the gain in the accuracy was more than 20%. So now this particular thing, and why it was happening, now we had three different breeds, Gir, Sahiwal and Kankraj. For Kankraj, number of animals were only 500. Whereas for Gir and Sahiwal, it was more than 1200. Then there is another problem, like strategies for GVV prediction when we have a huge number of animals and then HF inheritance, like it varies a lot. And now for this particular problem, we have started working. We are using a lot of other approaches, and we are planning like how to account for this particular differential genetic background in the prediction skills. So then there is again one more thing: can we incorporate novel traits in our genetic evaluation system? It's difficult, but it is possible. For fatty acid profiling, vaccine response, hemoccus contractors, yes, we can do that because genetic parameters are available. Heat tolerance, climate resilience, yes, we can still do that. But then there are only some traits with genome-only trait, like MIR spectra, workability, methane emission. So I have a few suggestions, like for existing breeding policies, we need to keep in mind smallholder production system in crossbred as well as in indigenous cattle evaluation program. Enhancing selection intensities and incorporation of the fitness trait, as everybody is talking, is very, very important. Addition of real-time genetic parameters in BLUP is very, very important in crossbred cattle. Why it is important, we'll come to that very soon. For indigenous cattle, yes, again, accurate modeling for missing pedigree, holes in the pedigree, loose structure derivative system is important. Again, strong record keeping along with like pedigree records is important, and integrating AICRP or network herds in the National Genetic Evaluation Program is really important. So again, when we go for modeling for increased dimensionality, as the topic also talks about future implications, with genomics, data dimensionality will increase fold. And we know that intensive selection reduces genetic variance, so obviously heritability will also decrease. Now, if we don't have accurate estimates of the heritability, your responses are going to be biased. Your prediction of the breeding value is going to be biased. So we need real-time genetic parameters which are essential in genomic selection scenarios. Now, how to estimate accurate genetic parameters? We know there are methods like REML, GREML, single-step GREML, and deep sampling, but with huge data, it's impossible to get estimates with any of them. Again, with impact of intense genomic selection, even if you select only for production trait, your genetic parameters are going to change. That we could show in our study. So to address this, we could come out with a new approach to get genetic parameters for large genomic data, and this particular approach estimates heritabilities and also genetic correlations in real time, even if your genetic parameters are changing every generation. And why it is important? It is important because if you are estimating your heritability as 0.4 with some other method, but if it is not 0.4 in reality, then probably your response is more, and then your boss may expect something more, and that will not come into the real picture. So these are the formulas that we could develop for heritability and genetic correlation. And good thing is, if REML takes 40 hours to estimate something with 40,000 animals, this takes hardly half an hour. So if, say for example, 100,000 animals, again, it takes hardly 8 hours. So now this is what we could show, heritability for production trait. This is the realized heritability over 10 generations. With pedigree, estimates were biased. Why? Because we all know REML will look at the base population, so always heritability estimates will be higher. What happens with the formula for predictivity that we could generate was much closer to the real estimates. Similarly for the fitness traits, and we also tested this method on extremes like 5,000 animals per generation and 100,000 animals per generation. And it was 3 generations clubbed together, so 300,000 animals per generation. And we could see that this method was really very well at both the extremes. For genetic correlation, again, yes, it works really good. Whether it works really good for real data, yes, it also works very good for real data. We could show that estimated genetic parameters for growth and carcass traits in Angus cattle were really predicted very well. So we have some future-ready methods. APY is already known to everyone for breeding value prediction, but now we also have genetic parameters by predictivity GPP for large data genetic parameter estimations using genomics. And Again, with this we have a small note on production traits, because recently there was a paper by Biswa and Lorenko. They have reviewed a lot of studies that were carried out mostly in the developed worlds for like increasing the production, maybe for meat industry or for the milk industry. And they showed that there are several detrimental effects on fitness traits which were observed because of intensive selection for the production traits. We did one study, the paper is in revision, and we could show that even if you select only for one trait, other traits automatically gets deteriorated. So intensive buildup of the negative genetic correlation between production and fitness traits occurs, even if there is no selection against fitness. And why this happens is because of the resource allocation theory, because genetically we have resources. It's not about the food resource. It's about the resources that every individual has inside its body. Like a cow can produce too much of milk, but there will not be much fat on her body. So resources will be allocated. So what do we want? We want optimum selection, especially in the conditions, because as somebody has already pointed out, we cannot send our cows for slaughter after 2 or 3 generations. So to conclude, Indian dairy breeding industry needs in-house solutions to address systemic problems. Indigenous breeds needs special attention and efforts to include them in national genomic evaluation system. With faster pace, genetic parameters may change very fast, and new methods for large to very large genomic data evaluation are already available. And then, with all this, definitely if we are looking forward for implementation of the genomic selection in Indian dairy industry, we need to go for one index. I strongly disagree with 15 to 24 traits, because we are not recording for traits. We are not at all recording for fitness traits. We have been recording for milk and then milk parameters, and 1 or 2 traits in very few selected individuals for fitness. So we need to include at least one fitness trait and then go for a genomic selection index, so that we can select best individuals and produce the next generation of individuals which are like optimum for our environment. So I end my presentation with thanks to all the research partners and some students who did a lot of hard work with us. Thank you.
Thank you, President, for the time. We're already on past time, so very quick question. I will take only one because we are short of time. Mike, please.
Dr. Gopal, one session and one question. If you have developed a model for estimation of variation for a smallholders population, as you suggested, 2 to 5 animals are kept in a smallholder system, then you try this model in resource-rich states like Punjab, Haryana, and also simultaneously in resource-poor states like Bihar or any region. So you can find a gene-to interaction, because productivity also depends on resources available to the animal. The robustness of your system, of your model, may be understood, that whether it works for all the system or whether it is working for a system which is resource poor or resource rich, because animals are also being kept in diversified situation in India. Then second point is that, or it's a question to you, at other platform also I have asked. Situation arises when GEB or breeding value estimated through progeny testing differs. If progeny testing breeding value, then which is the ideal parameter for selection, whether it is a GEB more reliable or progeny tested BB is more reliable. So those tricky things are coming all the time. I have encountered such situation in Bihar also when discussing the Pune semen station, and most of the time the bureaucrats, they rest on GEB. So this is
Interact with Dr. Saxena.
Okay.
So these questions are very useful. Kindly.
But if I take only 10 seconds. 10 seconds. So for second question, I'll respond. First question we will discuss. So I think whenever we are looking at the breeding values, so it's already proven on the theory and practical notes many times that genomically estimated breeding values, even for the newly born animal, are as good as progeny tested bull's breeding values. And then what should we prefer? We should look at the breeding values, maybe they're positive or negative values, but unless and until we don't look at accuracy and reliability, we should not believe the breeding values. If reliability or like accuracy is not 40%, and even breeding value is plus 1000, it's not sellable. That's what I.
Thank you. So thank you, and we will switch over to the next, Dr. Nilesh. So I think we have a time constraint, so I request you.
I'll be quick. And thank you, Alfred and Dr. Gopal, to explain what is genomic selection and how it is done. So I will be sticking to practical utility of what we have been doing with genomic selection. So this photo I have put purposefully. You see very ugly calf, but it's superstar for me, and I will explain why. I will deal with this. So Dr. Gopal told this equation is required to be taken in mind when you form some program for improvement, whether it is milk or fertility or anything, it is driven by this. And there are 4 paths of selection. So we select bulls to sire a bull, or dam to sire a bull, or we select AI bull to sire a cow, or dam which gives birth to heifers. So these 4, but in India, this approach, like this last path, because we are in smallholder condition, we can't cull cows, every cow has to reproduce. So this path doesn't work. So we have to take care of other paths. And in our breeding program, we try to keep this in mind. So we focus on selection of AI bulls. For that, we have to work on these 2 paths, and that AI bull will sire the cow. So 3 paths are taken care of. And in respect to breeder's equation, if you see our situation, when we want to select sire of a bull, we have these problems. Moderate intensity, because we don't have many proven or many bulls which have breeding values and so on. Accuracies are less for most of the bulls, because they were selected based on dams production record. And I still can't understand, even within our faculty, the affinity or the love for dams production record, which is very low or even lower, because many times it is predicted based on one-time competition record. So it's not even lactation record. And then we select bull based on this low criteria. Or sometimes we have some progeny testing programs, we get proven bulls. Generation interval is high. If it is dam-based selection, then 5 years. Otherwise, 10 years if we wait for proven bull, so bull may not be available. Only semen doses are available. So many people say that we should use proven bull for AI in India. Okay. So in dam side also, with lack of systematic recording, you have less intensity of selection, because very few recorded cows are available to select upon, and then very less accuracy, because you cannot correct for many of the effects. So your models itself will not work robustly, and then your accuracies are less. In generation interval, because you are waiting for peak production of that cow, maybe 3rd lactation, 4th lactation. So your generation interval is also very high. So these are the drivers of genetic improvement in our country. So if you put these aspects in your breeder's equation, then there will be a problem. In your breeder equation, if you just try just as a naive student of genetics, if you put this data, then you get this kind of situation. And this practically is not usable. And even if you use, because you take 10 years to prove a bull, it will have very less gain per generation. So what? And in this situation, we have to do genetic improvement of our breeds, and a lot of breeds, not 1 or 2 breeds. So we at NDDB, or we at national level with Department of Animal Husbandry, tried to put up a system which should work in these conditions. And then we started some progeny testing program or some pedigree selection program. Essentially, these programs work in a similar manner in each native breeding tracts. So I'm just describing those. We take whatever bulls are available for semen production in semen stations, and take some 15 to 50 bulls based on capacity of our AI network, and put them for test insemination. So village level, farmers are keeping cows, we ear tag them, record, and then put inseminations. do inseminations, record everything in digital mode, not on registers. On national database, right now it is Bharat Pashudan, previously we used enough. So our AI technicians go and record everything. Calf is born, if it is female calf, we apply again ear tag, record it, monitor it for growth. When it's cow, person goes and records that cow. While recording, morning, we do test day recording, 10 records in a lactation, and we take sample. That sample is tested for milk components, at least fat, protein, solid not fats. We have all reproduction data, so we have reproduction traits also with us. So these are the basic material which we work on. Whatever I am explaining is for milk, but it is true for fat, protein, SNF, and age at first coming and days open. So these parameters are evaluated in same style, and then you can extrapolate things. One of the best thing which did was strict supervision. We don't want incorrect record. If I have a target of 100 records, I will be happy with 50 records, but accurate records. So that was the key in our programs. And then we rank bulls, top bull, top cow, nominate them, avoid inbreeding, and produce male calves. Screen for disease, screen for breed characters, because we are passionate. Murrah should not have white spot. So we screen for everything. And then rest of the male calves which are passing these all filters, they are supplied to semen stations. And with this process, we on an average get accuracy of 55%, because we have high proven bulls accuracy and dams are recorded, so we get this kind of accuracy for these young bulls. And till 3-4 years before or 5 years before, we used to give these bulls to semen stations for semen production across the country. These are the programs across the country, and this is what we expect from these bulls. Just simple putting data in your breeder's equation. This is what we expect. Then came genomic era. We were excited, oh, genomic is coming, but it was very difficult to implement in our condition, because we never thought I should take DNA from this cow which is recorded. So in 2016, we started thinking, and then we started collecting DNA from these cows. Simple logic. If you don't have pedigree, it works like this. You have reference, you have genotypes. Compare your selection candidate where it matches. Very simple terms. I'm not going mathematical modeling and everything. You estimate each SNP effect and all. No, just compare it and get what is the production potential. Sorry. Oh. So there are a lot of slides, yes. I have to be faster. So I don't want to explain everything what Gopal sir has already explained, that we use single-step genomic blood where you take care of pedigree as well as genotypes. So this is simple, and the best thing was I can select bull as soon as it is born, like pedigree selection, but breeding value has higher accuracy. So whatever progress I expect is more. Accuracy, many times people think that how to evaluate this. So if I have 10% accuracy, if I select 50 bulls, only 4 bulls are correctly selected, the rest are either under-selected or not selected. If I have 90% accuracy, I have 32 bulls correctly selected, rest are rejected or selected wrongly. So this is how accuracy plays. So it is nothing good, nothing bad. You have to choose a path which gives you maximum progress. So now we do genotyping of bulls in all the breeds. Like we are now 7 breeds applicable for this process. So we do genomic breeding value, purchase, and then supply to the semen station. So accuracy is increased. Our AI program is more robust. And these bull calves are supplied to across country, and because we were using very low accurate measure for selection of bull, I expect 3 times more accuracy in our indigenous breed specifically. Like Gir or Sahiwal, we never had proven bulls or pedigree, deep pedigree information. So this will give very good gain. And now I am explaining one pilot which we did in our Holstein crossbred, how to accelerate this now. So we tried to see how many bulls are there in the country and what is their genomic breeding values right now. And then we thought, okay, if I record 5,000 cows and if I select top 500 cows, I can produce sufficient bulls which meets demand of the country. And from proven bulls, I have historical data and everything. If I take top 10 bulls, this is the kind of equation it will work, and then I will have good selection intensities, and then that selection intensity I converted into a cutoff breeding value for bulls. And then we decided in our projects, we will take bulls which are having 400 for more genomic breeding value. And we started screening all the young bull calves which we produced, and then selected those kind of bulls. So current scenario is this. This orange color bar is existing bulls average, and we tried to select this green color bar or above. So now we are trying to push average breeding value of bulls in semen stations across the country. So this is Holstein crossbred scenario. Similar is Jersey crossbreds. Mehsana buffaloes are very important to India because 50% milk is from buffaloes. So we have selected top 2 breeds, Mehsana and Murrah, for this genomic selection project where we were having programs. Murrah, I have huge demand for bulls, so my selection differential will be still smaller because I have to cater to semen demand across the country. These are Sahiwal, Gir. Demand was high up to last 2 years, before last 2 years. Now it has reduced, so we will try to push these levels higher. Now we were working like IVF have used in world for decades, and our research institutes were doing wonderful results, but they were one-off results. Then Government of India tried to push IVF for heifer production. But it won't work because farmer will not afford. So we thought, let us use this IVF technology for bull production. And that is the thing which, to our understanding, that IVF should be guided towards that goal. So what we did, like this is our IVF cycle. I don't want to go to details of this. This is the IVF cycle. So what we did, let us say instead of 500 cows, I use top 20 cows for bull production. And instead of all those 10 proven bulls, I use top 4 bulls. Of course, people here will say, what about inbreeding? I don't mind little inbreeding building up in my population as far as I am progressing in desirable traits. And Holstein has done that, Jersey has done that. Right now, I am not worried. Maybe next 2-3 decades we will not worry. After that, we may think of inbreeding. Because it's not very small population. We are working on 300 million animals. So it's not question of inbreeding. So we did this and we thought now we can collect bull cows with 700 or more breeding value. So think about selection differential and think about progress in next generation. So this pilot we did in our Holstein herd. This is now superstar for me. 900 breeding value is among top 0.25% of bull calves in this country. So this is the bull calf which was first bull calf born out of this experiment. And this is our initial progress of this pilot. And now we want to scale up this pilot in our national breeding program. We are linking now each IVF lab with some of our bull production programs to give them tasks. And they will give this kind of bull calves to the country. And hopefully with this we will progress faster. And again we are advocating DHD to include an index rather than only milk production, because initial our breeding objectives were like our document which says semen station can keep this kind of bulls. It was saying mother's yield should be more than this. Now that document says, okay, wherever there is a breeding value available, forget about mother's milk, you choose on breeding value, genomic breeding value. Now what we are saying is not genomic breeding value for milk, we are authenticating, choose on this index value which includes milk, fat, protein, age at first calving, days open to start with. And if possible, if we are able to successfully record things, then we include mastitis incidence, and gradually we include disease, all other disease, and then heat tolerance, and maybe in future feed efficiency, but that's distant date, but these 5-6 traits we want to start with. So these are the things, and these are the 17 initial bulls which we evaluated. Most of them are above 700 breeding value, few we will be rejecting. And this is how we want to go ahead in crossbred and in other breeds also. So a lot of people has contributed to this one, so I want to acknowledge. And our programs right now produce some 800 bull calves per year to supply to semen station across India. Thank you.
Thank you, Les, for nice presentation. A quick comment. We are already late.
Hello. Dr. Nilesh, in one of your slides, you have shown that out of 5,000 females dam, you selected only 40, giving a selected population percentage of 0.4. And then out of 200 bulls, only 4 bulls selected, giving a population percent selected only 2%. In this way, the selection intensities were higher in females and lower in male. However, conventionally, the selection intensity is very, very high in males rather than females. I surprise, or rather I worry, that such a schedule will lead to high amount of inbreeding in the further population, because number of males is already less and females is all you have reduced to such an extent. So that will lead to high amount of inbreeding in further population generations.
I would say, like, if I select, say 100 bull calves, only 50 are coming in production across, like, time horizon. 50 to 100, well, 50 to 70 are coming in production. So I will not worry at this stage about like maintaining big diversity, because breed-wise we have huge diversity. If we select some females with different kind of characters, it won't make much impact as of now.
No,
in future years, I have to worry.
Yes, just because you are suggesting a model, okay, so that model will go along for selecting making a, you see, path for selection of the breed as such. If people follow such a model, either the population should be such large that number of, because while you are doing selection, so many things you have to keep in mind. Inbreeding is one such parameter that you have to keep in mind. Otherwise, all your selection work will vanish due to inbreeding. So, because of that, you have to have a sufficient size of population, male and female ratio. Otherwise, this breeding program will not sustain. This is the worry. So, such a model will not be very, very, you see, successful if taken into a breed also. Because ultimately the ratio will remain same. You are working on the ratios, not on the population or breed. Ratio will be the same.
Sir, if you broad-base
my suggestion to you, or whatever you are doing, try to maintain effective population size to such an extent that inbreeding should not go beyond limits.
Yes, sir.
That is the
I hope the point, Dr. Les, the point raised by Dr. Saxena, you take care. Okay, so thank you so much. We have now an online presentation. Dr. Mintu Nath is joining from University of Aberfan. May please be connected.
Hello.
Yeah.
Hello.
Good afternoon from here. Can you listen us? Yeah, please. Yes. Dr. Nath, can you hear us? No. There is no audio.
I hope everyone can hear me.
Yes, yes, we can hear you. Please go ahead. Can you hear us?
Okay, thank you. Sorry for this technical glitch at the last moment. I thank the organizing committee for the invitations, and I'm really delighted to be presenting in this conference, and albeit virtually, of course, and of course, meeting all my friends and colleagues. So after these three excellent talks that we have just now, and I think they have all gave a fantastic view of the applied and practical aspects of the next generation breeding and integrating genomics and phenomics. So I'll take rather a more pedagogical view with some perspective from the medical science, and hopefully that will give some understanding about the overall theme. So here I'll just give an outline of the talk. I'll start with first the concept of the causal associations, and then of course the modeling approach that we take for estimating that, and then Mendelian randomization, particularly I'll discuss a bit of the procedures and how we can apply, and then some implementation framework. So why we need causality? So just this a very few maybe important reasons. First thing, of course, from the medical perspective, medical science perspective, risk assessment is quite important. What is happening in the future is quite important, and we are always interested about knowing that. So identifying the traits which are causally associated with outcome. I'll give a very simple example. Let's say if it is smoking, whether it is associated with the cardiovascular disease. That's quite important to know. And from even the important from the animal point of view also. Animal science point of view, let's say define production traits, fertility traits, disease traits, consumption traits. And even from the management perspective also, it's quite important to understand the risk. And even from the management context also, we can take actions to minimize those risks. And then of course the second important thing to understand about causality is about understanding the marker trait associations, and that may lead to the identifying novel candidate genes, and also to even in medical science it happens quite a lot to understand the more therapeutic interventions. And of course again going back to the breeding goals from the animal science point of view, and which can help in the facilitating selections, identifying the multiple traits which could be important, and of course improving the accuracy. So here is called a directed acyclic curve, which is to give an understanding about how it works in most of the disease scenarios, or in any case when we are trying to establish causality. So we are interested about the exposure, and the exposure and the outcome. That's what the causality we're interested in. So the exposure is causally associated with the outcome. But in that process, of course, there are a lot of other things which could lead to this outcome. So again, coming back to the same examples, let's say exposure is the smoking, and outcome is the cardiovascular disease. And of course, cardiovascular disease may depend on other covariates like age, sex, and also the cardiovascular disease also, even for other than smoking, it may also be associated with some other cases which are called confounder, which is influencing both exposure and outcome. And one confounder, for example, alcohol consumption, sometimes when someone smokes, they also drinks alcohol. So alcohol also is an important confounder. And also there could be mediator. His exposure is mediating through the outcome, for example, BMI. So smoking may increase the BMI, and BMI may relate to the cardiovascular risk. So the mediator is another important thing. And also there could be moderator. Let's say exercise. Exercise can reduce or diminish the effect of the exposure, the smoking. So someone is doing heavy exercise or physical exercise, they may reduce the cardiovascular disease outcome. And then there's another scenario, it's called collider. which is the hospital admission, when exposure and outcome both are influenced in glider. So when these are the scenarios, it's quite complicated actually, how we should model it and what you should really include in the model and what you should exclude in the model. So in general, the standard approach is that we should always account for confounder and covariate and never should account for mediator because that will diminish the exposure. And the moderator we should include as some sort of introduction term in the model. And again, collider term we should not use. So if I just go back to the standard scenarios about the way the research studies get conducted, let's say most of the cases is like a cohort studies or case control studies where we take the data and then either prospectively or retrospectively, we try to follow how our outcome and exposure are causally associated. And this is generally represented by a very simple some sort of this linear model, like when you have this exposure and all those confounder, we adjust for, and that could be having some sort of error distributions. Here, of course, it's just I'm showing normal distributions, but this Gaussian distribution is not a limitation. You can have binomial distributions, or you can have a Poisson negative binomial, gamma distribution. So it could be any other distributions, but the modeling framework is exactly the same. And in case of animal model, we can go further. We can include about the animal effect, of course, that individual term, and that's the standard modeling framework, including where your U is distributed, having a Gaussian distributions with the A, the relationship matrix. And of course, if you want to address the genetic associations, we can do the multivariate block or multivariate animal model context, and where we take those two variables, Y1 and Y2, and then again, the standard framework is there to estimate the genetic correlations. But this modeling framework sometimes is also quite limited. For example, we may not have all details about every possible confounder or maybe all possible covariates. And more so, sometimes there may be incomplete recordings, there could be incorrect recordings, or even some cases, particularly in the medical context, we are sometimes interested about some of the medications and the medications positive presence or absence. That also cannot be sometimes possible from really establishing the exposure outcome, that causal associations. So what we can do? So generally, the one that is more evidence-based is the randomized control trial. So randomized control trial is actually that individuals, as we know, that all those patients are randomized to one of the arm, for example, medical interventions, and another not in medical intervention. So again, going back to the cardiovascular disease, let's say there is a medication which could improve the disease condition. So we can randomly allocate someone with that intervention group and someone not, and then of course we can see that how this intervention is happening. So that's the randomized control trial. But randomized control trial is really very expensive, and it's also sometimes for some of those it's time consuming, resource intensive. For example, in the UK, it takes around 2 to 5 million to run a simple randomized control trial to conduct. So, and also there are some ethical issues. For example, with the examples that I've given, you cannot have randomizations about the smoking and not smoking. That's just not possible. So there are some ethical issues there also. And that's where this Mendelian randomization concept is coming into place. So Mendelian randomization, theoretically, when we were born, is actually some of those alleles, effects alleles, and control alleles were randomly allocated to us. And we are just going through this whole life, that randomization process. So it's almost nature's way of conducting a randomized control trial. And then, of course, the confounders are equally represented between the groups. So it's almost making the randomized control trial, but it's almost like a nature's way of conducting it. So here, then how we can do that? So that means the genetic, or let's say here is a single nucleotide polymorphism of that marker that is associated with exposure, and then that exposure is associated with the outcome. So that means if an exposure marker is strongly connected, then we can conduct that. We can have the Mendelian randomizations, and we can see the presence and absence of those, how it is changing. But there's some sort of assumptions there for considering this randomized control trial. And these are generally called, so SNP or this marker is considered as a single nucleotide polymorphism. It's an instrument variable. And the one important assumption that this must be associated with exposure, because that's what we're interested in. But there's other things is that SNP or that marker, that variant, that instrument should not be associated with confounder, and that should not have any associations with the outcome variable. So that's the three basic assumptions that must be met before we can consider about the causal assumptions. So let's take this, what about the different MR methodologies? And so basically the approach is that we have the different instrument variables. So basically from the GWAS studies, we get all those instrument variables, and then we extract both instrument variables for exposure and outcome, and then harmonize exposure and outcomes. And then we basically that GWAS studies, standard GWAS studies, and that model that is being presented here. And then what we do is just to estimate the effects of the genetic variants on the exposure X, and then of course the genetic variants on the outcome Y. So that's what we do. And then it's basically very simple to get this for single variants is just the ratio of that. Of course, based on the individual studies or summary studies, it's slightly different in methodology, but essentially this is the standard methods. And you'll see the lots of the different methods being reported in the literature about the inverse variance weighted, maximum likelihood. Generally, the inverse variance weighted is the most common one. But then there's other methods available. And there's also recently more MR Eger, MR Presso. They're being presented for particularly checking many of these assumptions. As I mentioned, those are quite important. So let's give a quick examples here. So for example, we have conducted, I'm giving a medical example, so conducted a study on the adult height, the instrument variants, and how they're released to the cardiovascular diseases. And then we just checked several cardiovascular diseases, and then we also compared with the epidemiological scenarios without considering the variant, this name, and how this epidemiological model is different from the genetic model from the Mendelian randomization point of view. And as you can see, in many cases, they are very similar, but in some cases, they are quite different. For example, in some cases, we are finding epidemiologically, there's no evidence, but genetically, we are finding evidence. And this one, of course, is not really directly related to the Mendelian randomization, but what we did here, we just found that there are three variants which are almost mimicking how it is acting as a statin that many doctors prescribe for lowering the LDL cholesterol. And what actually can happen that basically those having those particular allele, that means from birth, we are almost like on a statin, those having those effect alleles. And then we can see that how those individuals having a long-term medication effect can contribute to the different health condition. Like for statin, having statin, let's say for whole lifetime, reduce the risk of cardiovascular disease, chronic heart disease, but of course it increases the risk of type 1 diabetes or type 2 diabetes. For different MR studies in cattle, there are a couple of, only very recently, a couple of publications on MR studies on meat production, on health, this genetic associations. There are some also on dual purpose, and also the conformation metabolisms and production rate. So there are some couple of literatures now coming into and which are presenting those different Mendelian randomization association studies for different exposures and production rates. So I'll just give a very quick overview in the next 4 or 5 minutes about how to implement that. Of course, it has not been, so let's say we establish that there's a causal relationship between this, some of our factors, some of our variables, some of our exposure to our important production traits. So how we can implement that? Of course, previous speakers already explained some of those modeling scenarios. So that's the genomic block, which is just having a SNP there, or those markers there, and we can just do a standard modeling, including those genomic block, which is this case is just having no real, means all SNPs are considered as equal here. And then of course single stage block where we can accommodate this both genotype and non-genotype individuals. which is just having exactly the same thing when we are considering the all individuals and also genotype individuals, and with that only difference is that H matrix is changing into the variance space. But then in that H matrix, of course that's the standard there, but in that G matrix we can have that exact diagonal matrix, and that can sort of control this how this different weightage will be changed. So that's the weighted single G block, and actually this is we fit with thinking about the equal weight, and then what we do, then back solve the SNP effects, and then iteratively we update the G, and then until that's stabilized. So that's the very standard process about the weighted single block. And actually, that's where it is possible to implement the genetic effect or the marker effect of this exposure and the causal association. And that's what, so that means think about that in diagonal. So one of the diagonal elements in the kth step of the iterations, you can have that additive effect of the marker, and they just multiply with this effects of the Mendelian randomization effect that we are getting after fitting that model. And of course, we can scale in some way to incorporate. So actually, we have a procedure in place which can just directly implement if we have a causal associations, and that we can implement here. So that's the frequentist way of thinking about weighting these effects of the marker for specific genes. And of course, as you know, that the Bayesian alphabets, the different methodologies also give quite a lot of nice interpretation and have more control about how we can control this genetic effect. So for example, Bayes A, which can think about that each variance having a different, each variant will have a different variance. So which is and having a sigma square Gj, so J allele. And then of course, each one will have a inverse chi-square distribution with some degrees of freedom scale effect. And then Bayes B is just is similar as Bayes A, but here it's not just everyone, all genes, but all SNPs or all markers, but rather a proportion of that. So just most of them having 0, and the rest by some proportion of them following the same variance. And Bayes C, that's slightly a simpler version of Bayes B, when not having a very constant variance for all the genes, but again having that proportion, which is sometimes having a spike for some of these genes, and rather for most of the genes, and for some of the genes having a broader understanding. So that means we can, let's say we have the
Sorry to intervene, Dr. Nath.
We have identified
You have 1 minute
production
left.
traits, and then interested about disease traits, reproductive traits, conformation traits, and there's other SNPs. And we have, let's say G1, G2, G3, we have identified some causal associations. So those we want to weight more in our genomic selection process. And the other SNPs which we can integrate as the standard genomic selection. So that means we can just implement some Bayesian factors having the Bayes A component for those who are causally associated, giving more weightage on that. And when the other SNPs are having the standard Bayes C, and we can so that means we can integrate both of these components in our genomic selection process. And I've just done a very simple some simulation studies. As you can see, that this correlation between the true value and expected what is predicted value are actually very close. And of course, the fourth one, there you can see the correlations are slightly lower. So I'll just end very quickly. So as I mentioned in my presentation, the causal association is quite important, and particularly can establish the vertical theotropy and give better genetic insights, new selection targets, and of course the reduced confirming informed breeding decisions and integrated modeling framework is currently available. And then most importantly, that means what we need is a large-scale GWAS and then of course more exposure evaluations and the population structure. And having those large-scale data, of course, available to researchers across the world, and having this more open data access, like the biobank that we have for medical data. So I think that can accelerate the whole process and then establish the genetic associations, and of course, accelerating the genomic selection process. Thank you very much for your attention, and I'm sorry, I can't hear much, so possibly
Thank you,
questions.
Nath, thank you, and
Thank you.
appreciate it, right? So I hope there is communication gap also between there and you. Well, thank you, and thank you so much. So we will move on to the oral presentation, that is best abstract presentation. There are two One is by Dr. Rani Alex. You have less than 10 minutes. 8 minutes, right? You have 8 minutes.
Very good afternoon to one and all present here. Myself, Dr. Annie Alex, presenting on behalf of the Animal Breeding and Genetics Division, ICAR on the topic Implementation of Genomic Selection in Indigenous Sahiwal Cattle at ICAR Karnal. So moving on, since 2001, when Muson has introduced the term genomic selection, it has been the buzzword for the animal breeding, livestock breeding, as well as in the various agricultural sector. And it is genomic selection is a form of marker-assisted selection in which the markers covering the entire genome are used for the, so that the QTL, at least in linkage vesicularium, will be captured the marker information. But in the various national and international level, it has been implemented in the genomic selection in various developed countries since 2008. This is the sum of the developed countries. It has been already implemented, and since 2019, these are the 7 continents where maximum genomic information has been collected. But coming to the India, we have major challenges in the genomic selection where we have inadequate reference population, poor and phenotypic recording and data quality. These are the 2 major challenges we face when coming to the implementation of genomic selection in any indigenous cattle, since we don't have established progeny testing programs anywhere in India. So what may be the solution when we thought of it as utilize the already established breed-specific nuclear herds where the data recording programs has been going on. So this information will help us to capture extra information in the case of pedigree, by use of pedigree to improve the accuracy of the breeding program. So in genomic selection, I take accuracy as the keyword so that we can have an efficient implementation of the genomic selection. So this was our study in which we have used the history and the data sheets which was recorded for the last 70 years, where we have complete information of all the pedigree and the phenotypic records. We have further used the recent generations from 2000 onwards, whatever the DNA storage in our labs, and we have used 503 animals. We have genotyped it and quality control it. We have standardized the models for the perfect accuracy evaluation. Then we have cross-validated the models. So this was our Sahiwal reference population. It was in the over a span of 70 years. We have total number of 1,900 animals, almost 2,000 animals. And this is the list of data of last 7 generations. We have used this information to the implementation of the genomic selection. First, we have carried out the least square analysis where the overall production was almost 1,500 kg. And further, we have used the genotyping filtering where we have removed the missing information, hard event break and minor allele frequency. And we have obtained almost 30,000 SNVs which are in the QC for the downstream analysis. Further, we have standardized the model suitable for our Indian conditions where we have used year of birth, season of calving, and year of AFC as co-variable. And we have estimated the genomic heritability, and it was almost 0.17 in the Sahiwal population in the NTRA herd, which is very good. And further, this is a strategy which we finally followed in the genomic selection program in the NTRA herd, where we have used the pedigree population, where we have further genotyped the recent generations of animal, and further we have used this information for the selection of the young bulls. Using SSG method, whether we have blended both pedigree and genomic relationship metrics. So this was the final breeding ranks which we have obtained for all the bulls. You can see that the ranks, top ranking animals have a plus 300 kg genomic estimated breeding value, which we compared with the EPD, which was used, normally we have used as a criteria for the bull selection. And our EPD always also matching with this, the GBVs. And further, we have excluded some animals from the breeding program where we have got almost negative or almost zero GBVs. So these were the top breeding bulls we have selected and inducted into the breeding program. First one was almost with 300 GBV and with an accuracy of 0.35, and second one was 289 and with an accuracy of 0.37. These were the some of the animals which were top animals we have selected, and some of the animals which we have excluded from the unheard. So concluding, we have established a genomic evaluation pipeline for dairy animals and generated the baseline information and identified allied bulls. So the NDRA has implemented genomic selection program in the Sahiwal cows, and what we are looking for the way forward is the inclusion of other ASCRP herds for the maximum increasing the accuracy. And the second one, as we are discussing throughout the day, including fitness traits and other traits, and develop a genomic selection index for using both production, reproduction and climate resilience. And it can be taken as a first step towards the precision animal breeding, where the next Gen 5 we are expecting using deep genomics, machine learning, and where we have giving weightage to sustainability and One Health program. So next Gen 5 program where we have more precise, efficient and sustainable breeding program. Thank you all.
Thank you for praising before time. Any quick comment? Anybody? This is selected as the best abstract for this technical session by a committee. Okay, so if there is none, thank you.
I wanted to ask a question, sir.
Yes, yes.
Can you please comment exclusively? This side, on your left. Yeah.
Yes, sir.
Yeah. Can you please comment on the accuracy of your selection and how does it stand with your traditional progeny tested ranking versus your genomic?
Sir, actually, Gopal sir already told you when we estimate the GBV, it is equivalent to the progeny tested program.
Okay.
So what we got the accuracy is almost total accuracy or average accuracy of the herd we obtained almost 43%. It is well and good when we are taking the reference population of 500 animals with genotype information.
Thank you.
Okay, thank you. Thank you, Dr. Rani. I'll invite the next Professor Ashish Kumar Bose.
Good afternoon. Respected chairman, reporter, and the dignitaries present here. My topic is expression profile. That means the functional characterizations of the Ebola DRB3 gene conjugate. Here the MSC molecule, you know all that it regulates the immune response, activating the T cells, and it is more polymorphic, and the region of the MSC, the BOLA, which is regulated the bovine mastitis, and we say the polymorphic site is class 2 gene, and if we say the bovine leukocyte antigen is a part of MSC, and DRB3 gene, this encodes the glycoprotein present in the peptides of the CD4, and the CD4 is the surface protein which present in the T cell, and that regulates the immune response. And also it regulates the disease resistance. You can say the MSC molecule present located in the 23 chromosome in the Q region 21, and because of its high polymorphism, it serves as a candidate gene for association studies and with diseases and immunological traits. The main objective is to characterize Ebola DRB3 allele in crossbred cattle and to analyze their predicted protein structure, functional domain, and antigen binding potential using bioinformatory tools. The aim was primarily the characterization in silico method provide rapid, cost-effective insights into protein properties before experimental validation. We have taken 200 crossbred cattle, GBP, maintained at GBP for the University of Agriculture and Technology. We have done the PCR amplification, taking the specific primer of DRB 3.2 gene, isolated DNA, and then PCR amplification and the sequencing studies. And uses the bioinformatic tools, the nucleotides and derived amino acid sequences are aligned with the sequence specific, breed specific, the cluster W method Magaline program, and the disparity index is measured to observe the difference, the substitution pattern for a pair of sequences. The composition distance is measured to the differences of nucleotide compositions of the given pair of sequences. The bioinformatic analysis is done taking the intrapro scan for identification of the functional domain. Swiss model workshop to know the 3D structure predictions, RESMO software visualization of the protein structure, and the ProtPram software knowing the physical chemical structure properties of the protein. The IEDB, that is MSc class 2 binding prediction and epitope identification. So for in silico characterization, several bioinformatics tools were used to analyze protein domain, the three-dimensional structure and intrinsic properties. This helped to infer the functional potential of each allele associated with the clinical mastitis in the by taking the affected animals and this using the chi-square test. And we know taking the alleles, we have the 6 alleles identified, and from this the allelic diversity is mentioned. We have here the functional mutations are there in different positions of nucleoside as well as amino acid sequences. And we have known that the E allele having the maximum functional mutations, and followed by the B, F, A, C, and D allele. And we have these 6 alleles having the accession numbers. And the disparity index is found. It varied from 0 to 1. So it gives you testing the homogeneity of the substitution pattern between the molecular sequences. The composite distance is a measure of differences in nucleotides compositions for a given pair of sequences. This is calculated, that is the half of the sum of the square differences in counts of bases. And the software mega forced computers, this is used for calculating the composite distance. These are the amino acid sequences of the proteins, and I have shown you this. This is showing the amino acid residue substitutions, and the maximum 4 substitutions occur, that varies from 0 to 4. Somewhere there is one single nucleotide substitution, somewhere 2 nucleotide substitution, amino acid substitution, somewhere 3 amino acid substitution, and somewhere 4 amino acid substitution. So by taking this ProtPram analysis, we have this protein domain identification here, that is IPR 0035. Yes. This corresponds to MSc class 2 glycoprotein transmembrane antigen precursors of signal beta chain. And these 3 modeling revealed the conjugate peptide binding group with allelic variations in side chain conformation. This is the 3-dimensional structure of the protein, and this is visualized by RESMO software. So the physicochemical structure of the protein is encoded by the gene, that is using this ProtPram bioinformatic tools. We have different amino acid, the protein properties, the physicochemical properties, the instability index, hydrophobicity, amino acid composition, extinction coefficient, and the PI, aliphatic index, isoelectric pH.
Conclusion slide, less than
Yeah.
45 seconds.
And this protopharm analysis revealed that glutamic acid has the most frequent residue, possibly influencing protein changes and antigenic presenting efficiency. And this epitope predictions through IEDB allowed us to identify potential antigen binding regions for each allele. A high consistent rank indicates the allele with better antigenic presentation capability. And association, we have found significant association is found with mastitis, and this in the order of A, B, A, D, A, E, B, C, and C, D genotypes. And concluding that the present study revealed that high allelic diversity is found in this gene. The allelic differences may influence protein stability and immune potential, provides insight into marker selection for disease resistance. These findings can help to identify allele associated with superior immune response and disease resistance states. Thank you.
Thank you. Anybody has any comments? No. If not, thank you. And I think we have come to this technical session 1, and we have passed already 30 minutes late running. We are supposed to finish at 3:30. So with that, I think we had four, my colleague, Chairperson Dr. Trivinder, Dr. Subodh Kumar, Head and Genetics. We are thankful to the organizers to giving us this opportunity to finish. We started at 2:20 and we are almost only 10 minutes more than the session assigned to us. So I think I will hand over now to the organizers to do the further.
Thank you so much, sir. We are going to start now to carry over our love and gratitude to our speakers as also our chairpersons. So I request Dr. Biswa to hand over a token of love and sincerity. Please come forward. Dr. Alfred De Vries. Dr. Alfred De Vries. Please come over the dais. I request now Dr. Misra to hand over the memento to Dr. De Vries. Dr. Gopal Gawani, sir, please come. Dr. Gopal Gawani. I now request Dr. Misra to hand over the memento to Dr. Gopal Gawani. He is not here. Dr. Nilesh Nai. You have to adjust time. Dr. Nilesh Nai. Since Dr. Mintunath is online, so we can hand over. Dr. Rani Alex. Dr. Rani Alex, please come over the dais. Where is he? Dr. Rani Alex. Professor Ashish Kumar Ghosh. Dr. Misra, please come. Dr. Subodh. I request Professor Punendu Biswa to come up the dais to hand over memento to Dr. Misra. Now I request Professor Punendu Biswa, sir, former Vice Chancellor of University. He will hand over the memento to our chairperson as also the podium. Dr. Trivedi, please. He is not here. लेके जाना chairperson. So Dr. Subodh, go fast. Time is not there. Abhi de do. So thank you very much, sir. Thank you very much, sir. Now we are going to start successively now technical session 2. I call Chairperson Dr. Vineet Vashin, Dr. Tarun Kumar Bhattacharya, Dr. Vikas Bora. Please come. Dr. Vineet Vashin, Dr. Tarun Kumar Bhattacharya, Dr. Vikas Bora. Report here Dr. Amit Kumar, Dr. Aruna Pal, Dr. Amit Kumar and Dr. Aruna Pal. Now chairpersons will be Dr. Vineet Vashin, former principal scientist, Animal Genetics and Breeding, ICAR Headquarters, New Delhi. Dr. Tarun Kumar Bhattacharya, Director, ICAR NRCE, Hisar, and Dr. Vikas Bora, Principal Scientist and Head, AGB Department Division, ICAR NDRI Karnal. And rapporteur will be Dr. Amit Kumar, Principal Scientist, Department of Animal Genetics, ICAR IVR Ijjatnagar, Dr. Aruna Pal, Associate Professor, LFC, Livestock Farm Complex and WBUAFS. In the next session, we are doing Dr. Sarani, please. So I now hand over the mic to our chairpersons to lead the session. Dr. Vineet Vashin, Dr. Tarun Kumar Bhattacharya, Dr. Vikas Bora will be our chairpersons, and rapporteurs Dr. Amit Kumar and Dr. Aruna Pal. Dr. Vineet Vashin, please, sir. Dr. Amit Kumar is here. Okay, sir, we can start. This mic is working or not working? Mic is not covered. Mic.
Good afternoon, everyone. So, in this session, we have hello. Okay. Hello. Yeah, that's fine. So, in this session, we have 4 lead speakers, lead papers to be presented. And the time allocated is 20 minutes to each. I hope they stick to the time. Okay, it is 18 minutes, the revised timing. And along with this, we have two best abstracts, 8 minutes each. Okay. So I will now invite the first speaker, Dr. Srinivas Jagirdar, Principal Scientist, Fish, Genetics and Biotechnology, ICAR SIFE, Mumbai. Yeah.
Thank you, sir. Good afternoon, and respected chair, rapporteurs, and all the distinguished scientists. I am really out of water here because the topic that has been given to me was really challenging one because in aquaculture, initially what I thought is we are following the animal genetics, not only inspired, followed, or still following, but the things that when Dr. Datta called me and asked actually to take up this topic, I started wondering whether there is anything for me to actually contribute here. But when I started dwelling on that one, I found there are many things which are unique to the aquaculture genetics, and some of those things can be really taken up. So I have planned like this. Maybe we'll start with that. Well, this is actually just to show that one in India, actually the fisheries or aquaculture is not new. You can see that one mentioning in almost starting from Rigveda to the things. And okay, to something, and actually fisheries or aquaculture, in fisheries when we talk, there are two different types are coming. One is capture fisheries, another one is the environment. We have got a fresh water, we have got brackish water, we have got the sea water. Then again, depending upon the intensity of the culture, it is intensive, semi-intensive, extensive, and then different types, that is depending upon the feed, non-feed, fed, non-fed, all that types of things are there. To just simply give the definition, aquaculture is the controlled cultivation of the aquatic organisms like fish, shellfish, and as I mentioned, we have got actually in our tradition also aquaculture was there. And of course, aquaculture of the fish is the very vast biodiversity is there. More than 35,000 species have been identified, and still many to be identified. Almost around 250 are added every year to this one. Everybody is talking about the around 10 breeds, 20 breeds, 30 breeds. We have got actually in aquaculture around 580 species are cultivated across the globe. And in our own country, we have got almost around 45 different species are cultivated for aquaculture purpose. Of course, there are top 10% species are there which actually contribute significantly. Around 90% of the production from aquaculture comes from these top 10 species. In this one, actually the major ones are the carps. Carps contribute up to about 30 to 35%, and in India they are actually about 80%. They contribute to the total fish production. Then Indian fisheries and aquaculture, actually we are number 2 in the world, both in capture as well as the aquaculture. We are producing about 8% of the total global production comes from India, and we are actually, the production has increased in this one's in 13, 14. If you look at that one, at present we are producing about 195 million metric tons. So Indian fisheries, actually we have got the major carps, that is the rohu, katla, mrigal, and also some other species are cultivated. Some catfishes like the magu, they have been cultivated. Mostly the production or the export is depending upon the prawns. So if you look at the aquaculture growth and globally, it is expanding like anything, and by 2050 it is expected to reach around 400 million metric tons, and in India it is expected to reach around 140 million metric tons. So, economic as well as the food this one security, or the also nutrition security, because the 20% of the total animal protein, it comes from the this one, from the fish. If we look at the contribution to this one, it is around 150 billion, and you will be wondering, actually the fish is the highly traded food commodity in the world. So there is some people argue for the pulses also, but as per the FAO, it is the highly traded commodity. Of course, India is actually, if you are looking at the contribution of the aquaculture or the fisheries to the GDP, it is 1.2% to the total national GDP and around 7.28% to the agriculture GDP. Of course, we are growing it in much faster. Just to give you an idea, actually, if you look at the protein content, which is very much interested to the people, so we have got actually the, protein content is much more higher in the chicken than compared to us, but if you look at the carp, salmon and the prawn, they give much better or more or less equal quantity of the protein per 100 gram of edible portion. So this is about the cost of production. If you look at that, the milk is the cheaper, and rather the egg is the cheaper, and carp, and this carp actually, especially when I'm talking about the carp, this is actually feed based, so therefore the cost is more, where actually it is extensive or the non-fed species are there, it is actually the much lower, this one. So this is about the cost of production in 1 gram of protein. Then let's see, and prior to that one, if you look at the genetic selection programs, the record says in 1920 onwards it has started, but not much on that one. It is actually by the farmers and non-scientific way it was done. And we have actually progressed since 1970 onwards. Actually, the aquaculture genetics has taken a much more leap. And you name any genetic technologies or tools, they are all being used in aquaculture. So this is about that. Actually, the first genetic work or the significant work of the genetics in aquaculture in the initial stages was so the species hybridizations are going on in aquaculture and they have been exploited also. There 32 and to the very high numbers about 100. So this is what actually we manipulate the chromosome sets, we produce the triploids. Triploids are used commercially and they have been used worldwide. So that's what actually is the chromosome set manipulation. Then sex determination, this is again another one more challenge in the aquaculture. You name any type of sex determination, you will find the example in the aquaculture. So that is the one big where the lot of genetic diversity is there. And then we narrow down actually after conducting the common garden test and other things. So that's why actually the aquaculture genetics gives a lot of advantages and the returns. So I will skip the majority of these things. I will just directly go to the, just want to show actually one major problem with us in challenge is pedigree identification. Because at the birth, the fishes are very small. As they are so small, we cannot actually tag them. We cannot actually put any tag on them. So we clear them family-wise till they attain a taggable size. And that takes somewhere about 3 to 6 months. And it induces a lot of other non-genetic variations in that one. So this is what actually we are showing. These are some of the tanks where actually we put one full SIP family for rearing till they attain a taggable size. So this is actually the one way, and it increases the generation interval. So that is actually the things we usually use the pit tags, and these are the after pit tagging we use them in the communal rearing. These are all the way. This is actually maybe the one of the interesting thing for all the geneticists. It shows actually the genetic variation both between and within family variation, which indicates that there is a lot of additive genetic variation is present. So this is actually how the breeding values they come, typical this one. Of course, we have got actually one advantage, as I told, the heritability of the traits, especially the growth related traits in the majority of the aquaculture species are very high. So therefore actually the improvement is much faster. Of course, we also work on the dissemination and we are actually having the different types of systems. Okay, I'll come to the takeaway from the aquaculture genetics. Aquaculture genetics is inspired by the animal genetics, of course. We are followed and are following it also. However, over the period of time, we have actually, the aquaculture genetics has matured and it has created on its own. This one, we have got actually some aspects here. High reproduction rate, phenotyping is cheaper, evaluation in multi-station, multi-environment conditions, all these things. These are the few takeaways. If you look at that one, we have got the domestication and founder start wide, then narrow, then breeding system architecture, that is population greater than the families, then reproductive control, then phenotyping and platforms, phenotypic platforms, that is what challenge, sensor and scales. I'll just briefly give you about the domestication and founder starts. As I told, we deliberately start with a very large base population, bringing in the different geographical stocks, assembling them, breeding them, going for actually the common garden evaluation test, and then we narrow down on which to take, whether to mix them, go for the diallele crossing, all those things are taken from that one. So what is actually take home for the livestock is, that is the founders from multiple ecozones, they should be looked into, immediate DNA tagging has to be done, then common garden benchmarks has to be found out, then early run of homozygosity, then any, that is a very important thing, any surveillance, then cryobank and embryo, cryopreservation wherever feasible. Also the breeding system architecture, that depends upon the species, it somewhere starts from 6 months to 8 years.
Is it possible to reduce this?
Yeah, we can reduce, especially in some of the species where actually the rearing them communal means family-wise rearing is takes a long time. For example, in salmon, it takes a quite long time. In case of magur, it takes a lot of time. So we can reduce that one very considerably if we go for the genomic selection, especially when we take the genome information for pedigree construction.
So generation interval is defined as age of parents when the offspring are born.
Yeah. Same, but actually what happens is because of the rearing system, it gets extended. Okay. So that's what we can reduce that one considerably.
Thank you, sir.
Thank you. Thank you.
Sir, what is the heritability you have found out for weight gain
Weight gain, sir.
in family selection case?
Yeah, actually it is depending, but the review of this one, it says somewhere it is above 0.4. Even in our own case, we have found actually the heritability 0.6 by half-SIP method also.
Okay, thank you, Dr. Jagrita.
Thank you, sir. Thank you very much.
Now I'll invite the next presenter, Dr. Kutubuddin Mullah.
See, if I tell you what are the tools available to generate genetic variation, these are the 4 tools: JDFN, Talent, CRISPR-Cas9, and then comes next generation tool, which I will reveal to you. So you see, this Dan Hart is actually pioneered the Talent technology, and then CRISPR-Cas9 and Jennifer Doudna, and then I talk about some next generation tools. So before I go into that, a little introduction. This is how CRISPR-Cas9 works. Here is a postman called Cas9. It has 2 scissors in his pocket. And what you give to a postman, you give a genetic address to the postman, okay? So here is the address in the form of a guide RNA that you can customize for your need. Then you give that address to the postman. The postman takes the address and it goes to the genome, and then it finds where the similar address is located, and then the postman gets the signal that here my work is to be done. And then it will make a double strand break here with one scissor, one strand would be cut, and with another scissor, another strand would be cut. Next, this is how you can make a very, very precise double strand break wherever you want in a spartan 2 address to target one exon 1 and exon 2 to reduce plant height. So, exon 1 and another is on exon 2. And in normal NHEJ pathway during repair, the intervening region could be deleted in some of the population. And as we also have obtained mutant or 620 base pair exactly precisely deleted for expanding from exon 1 to exon 2. And this is known from a natural variation that was used during Green Revolution to reduce the height of a plant. So we have the similar kind of objective, and we have achieved with CRISPR-Cas9, where the tall plant has met short plant by using 2 guide RNA and deleting 620 base pair from exon 1 and exon 2. This is how the result looks like. The middle one is the wild type, and both the flanking one are the edited one, which has 620 base pair deletion, and that causes the reduction of height. And that also makes the problem in India is CRISPR-Cas9 IP is very complex, and we as an Indian does not have the freedom to operate for its commercial application. So that's why our lab always wanted to figure out any alternative things that could be used, and that India has an IP. So we have developed this TNPB mediated plant genome editing tool that could be used as an alternative to Cas9 and Cas12, and that is derived from this bacteria called Deinococcus radiodurans. And it acts almost similarly. It has also this PAM sequence, and you need to give address, like long RNA sequence, to this TNPB protein. And if you compare with Cas9 and Cas12, where Cas9 is like a football, like 1400 amino acid, and this TNPB is only 408 amino acid. It means it is 1/3 of the size of Cas9. So because of its smaller size, it also helps you to easy delivery into the eukaryotic system. So now it mainly, introduces deletion, so that a lot of deletion means frequency of frame shift mutation would be also higher. So if you see, these are a simple, demonstrating example where we have targeted chlorophyll biosynthetic genes, which creates green color in plant. And you can see with this TNPB tool, we could generate albino plant. It means the chlorophyll biosynthesis pathway activator, gene activator. It means what we have done, this TNPB was made a dead TNPB. Dead TNPB means it can go to the target, but it will not be able to make any cut. So if you give an address to the dead TNPB, it will go there and it will sit there. But what we have utilized, the purpose of this, we have fused here BP128 and 6X TAL sequence. That's a gene activator, transcription activator. When you fuse the transcription activator with the dead TNPB and you give the address to the dead TNPB, then it goes to the promoter, and because of the availability of the lot of activator in that promoter, now a gene which was expressing very minimal level, it will be activated in a higher level. of the presence of this activator component. And we just demonstrated into 3 or 4 genes, or we got at least 10-fold activation of a gene without changing any gene sequence. Now, this has been also deposited to AdGene. You know, AdGene is an NGO which distributes plasmids to everywhere. So our this TNPB are being used in 15 different international labs from USA, Canada, and China, Belgium, and 25 laboratories from India. This has been very good international news also. And recently we got a IP, Indian IP for this TNPB tool. So now we have this problem a little bit at least solved, that we can reduce the dependency on IP protected Cas9 and Cas12a. Now coming to the more exciting story. So precise editing, can you tell me, most of them are Indian audience, but I see some foreign audience also here. But this is Amitabh Bachchan playing the role of Oro, and Oro is suffering from a rare genetic disease called progeria. And you know what is the difference between Oro and Oro's friend? The difference is a single DNA letter. As you all know that this is an SNP in one of the gene, and because of this single wrong letter in Oro's body, Oro was dead at the age of 12. And this happens a lot of progeria affected children who do not survive more than 12 or 13 or 15. This is a case of plant where a plant is susceptible because of the presence of a C nucleotide. When it is replaced with a T, it becomes resistant. So now the next question is not to disrupt the gene sequence. Question, is there a way to rewrite genetic letters very precisely? Or if you want to cure Oro, you have to precisely rewrite A to G nucleotide. So how it works? So base editor is a technology by which you can do this kind of rewriting, and this is David Liu from MIT who has pioneered this technology, and you can do a lot of this kind of changes in the genome very, very precisely. So let me tell you very easily how it works in the genome. So if you see here, what is the problem in this code? Anybody? There is one problem, one letter problem. Now you have base editing tool, and you could correct that letter with the right letter, and then you get your phenotype back. This is how base editing works in the genome. Okay, now does it really work? So I'll give you very interesting examples. In our lab, what we have done, this is an existing base editor, the upper one, which is the lighter color bar, and the other two darker color bar represents the data from the base editor that we have developed, which has enhanced the efficiency almost, in many cases 3-fold, 4-fold efficiency of very precisely A to G editing. You can see A has been modified to a G nucleotide. So now let me give you a very, very interesting example. In many cases, a natural mutation, spontaneous mutation, if there is a single base change happens in exon 1 or exon 2, and it causes a premature stop codon. Stop codon should be present at the end of a coding sequence. But because of the mutation, if a premature stop codon happens at exon 1, then the whole protein would not be produced because it is a stop signal for the translational machinery. Then the protein would be truncated, okay? Now, because of this kind of premature stop codon generation in one of the rice variety, in one of the gene, the plant became blast susceptible. It's a disease susceptible. Now, we wondered whether we could correct this premature stop codon by applying base editing. So we have applied base editing. Sorry, I was too fast to keep my time. Yeah. So you can see here is the result. TAG has been modified to a CAG. So CAG codes for glutamine. It's not a stop codon. Now this gene is corrected. So disrupted gene is now corrected with base editing in plant, and as a result, it produces a disease-resistant protein in its full length, and it gives a blast disease-resistant phenotype. Now, next advanced technology called prime editing, which we also work and champion on, at least in our lab. We are very, very efficient in it. So this is an example how we can correlate. This Tay-Sachs disease is a genetic disease which is caused by this 4 extra DNA letter in hexagene in animal. So, and they die at the age of 5 only. Now, if I ask you, what is the hypothesis? You could cure this child from this disease. What is your hypothesis? Suppose you have all the ability to do in this world, and God has told you that, okay, you ask me anything, I would give you. What you would ask to cure this child? You want to delete this 4 extra nucleotide. Yes or no? If you can delete this 4 extra nucleotide, then this gene will be corrected. Now, this could be also done by this technological prime editing, and it is made up of a N-Cas9, it's a modified version of Cas9 and fused with a reverse transcriptase. And you can do all this kind of mutation very, very precisely as you wish. It's not a random thing. So here, if you see how it works actually, it has the guide RNA of the genetic address has two components. One is a search component, the lower one, PEG RNA. Search component tells where to edit, and the replace component tells you what to edit. It means the same address or genetic address tells you where to deliver the job and what to deliver. Both of them are coded in this PEG RNA. Now I will give you a simple example from our lab. So this is how primary editing can work. Here the code has little more problem, and you need to correct this code by deleting 3 letters and inserting 1 letter. Okay, so this is called complex editing. It's not a simple replacement. Now the primary editing could do the similar job by deleting 3 precisely and inserting 1. This kind of combination of edit is also possible. Now I'll give you an example of how evolution gave us an idea and what gave us a wonderful crop improvement application. You know, C3 photosynthesis and C4 photosynthesis. C4 photosynthesis is always superior than C3, but the rice that we eat today in lunch, they are all C3, they are inferior in photosynthesis. But the maize that you eat, they are all C4 photosynthesis. They do photosynthesis. So, but all enzymes and machinery are present in, all C4 enzyme and machinery are present in C3 plants. But when you compare two, one enzyme called PEPC, phosphoenol pyruvate carboxylase, you can see there is arginine, R for arginine in C3, which is replaced by glycine in C4. So this is single amino acid change in this enzyme when you compare from C4 and C3. And they say that this is important to make the enzyme more functional in presence of its inhibitor. Enzyme is inhibited by the presence of some chemical. But C4 version is less sensitive to the inhibitor, but C3 is more sensitive. And this is happened due to evolution. Now we wanted to mimic that, whether we can mimic that glycine in a rice Pepsi enzyme. So in order to do that, you need to make a CGC arginine, this is in rice, and you need to make C to G, so it becomes GGC, which is actually glycine. So we are not going for generating any transgenic plant, but we want to change that special arginine to glycine in C3 Pepsi. And we are successful by applying prime editing. You can see the C has become G. This is a real data Sanger chromatogram. So arginine became glycine in rice. So it means from evolution we have learned these mutation works and we have installed it in rice. And let us see what is the effect. Here is the wild type C3 rice and the mutated plant has darker leaf. It means it has more chlorophyll. And as a result of more chlorophyll, it also shows more photosynthesis. So photosynthesis and chlorophyll accumulation has been significantly enhanced by this single letter change, which was evolutionarily informed. And if you see here, interesting that the seed size in both length and breadth also increased. These are the 10 seeds comparison from wild type and the edited plants. And also 1,000 seeds weight, because the seed size is increased, the weight is also increased if you compare from normal and versus edited one. And this is the most exciting one. We never expected. It's an accidental discovery. So this seed also accumulate high iron content, high zinc content, and high protein content. And we named this rice as a zip rice, zinc, iron, and protein, which is highly desirable to avoid malnutrition. So this single nucleotide change has made tremendous effect in this plant. And now this is the last portion, one slide, and then I will finish my talk. So as this conference talks about artificial intelligence and machine learning, so artificial intelligence can be used to design a protein. So as I said in my beginning, that our lab has a focus to avoid or solve the problem that Indian researcher has, that is IP issue. Okay. So if an AI can design a protein, and if that protein could be used in genome editing, then it could overcome the IP barrier of global south. And we developed an editors based on AI designed Cas protein, which is 400 mutations, different, divergent from the naturally occurring Cas9. And if you
Dr.
see,
you have just 1 minute left.
yes,
Complete
yes,
your
I'm
presentation.
well on time. I'll finish before. Thank you, sir. So if you see here, Cas9 is the natural one which is derived from bacteria, but OC1 is the AI designed protein which is at par with Cas9. It means the artificially AI designed protein is working at par with the natural Cas9, and this could actually revolutionize this one. And we have developed a lot of different editor like base editor, prime editor using this Cas9, but I have not enough data to show you, but this is the beginning of that. And we recently, in this area, we are too much confused which computational tool to use, design our experiment and to also analyze the data. So this is a recently published, just October 31. This has become online. So we have made a repository where you can go and select which kind of experimentation you want to design, which kind of data you want to analyze, and you would be directed to that specific tools that you need to do to avoid, minimize the time requirement. So these are the 3 books that we have edited recently, and this has become very, very popular. If you want to apply in fish and animal, a lot of protocols are all there. And this is the time to acknowledge, this is my group, and these are the generous funding from Ignite, and I recently got another project from NSF US. So thank you very much, and because to remember Watson, this is James Watson, I was very lucky to meet him in Cold Spring Harbor Laboratory, because he was just few days ago, I think Professor Tikelot has mentioned during his presentation this morning. So it's a tribute to James Watson, because DNA discovery actually made all those kind of genetics and genomics things that we are using. And thank you, sir, for inviting me. I do not know what you have seen in me that you have invited as a plant scientist in this huge learned audiences of animal and fish genetics. And thank you very much, sir.
So, any questions or clarifications?
Yes, sir. Huge. I am helping one researcher from CFA Mumbai for fish, myostatin gene editing in chicken, Avian Research Institute, and also for cancer from Tata Cancer Research Institute. So I designed their technical part, and they do in transformation.
Who is in collaborating with you?
Oh, it's a Gautam Kalluri.
Gautam Kalluri.
Yeah, he's my friend. Yes, yes.
That was an excellent presentation.
Thank you.
So this IP issue with CRISPR-Cas, I hear a lot of times from colleagues that work with crops, but I never hear it in livestock. Why is this?
So, could you repeat the last sentence? Livestock?
So the IP issue with using CRISPR-Cas is a big thing in crops.
Oh,
but
yes.
in livestock, my colleagues never.
No, I think you see, livestock, genome editing in livestock is very, very limited in global south. It's not normally very good at UC Davis. The UC Davis team is very strong. So they have developed low methane emitting cows. So I think when the scientists from this region want to use those tools, they will obviously face the same issue. And why in plant I'm saying, because we have already developed 2 crops that are edited through genome editing technology, and our agri minister recently announced that we have developed this, proudly say that the 2 rice varieties. But yes, developed and it's identified as a variety, but it cannot go to the farmer's field until and unless the negotiation is done with the IP, and ICR is really helpless. It has been already 2 to 3 months, but it could not figure out what to do, how much money to give, so it's very difficult to negotiate. So it's not an easy thing, actually. Yeah.
Well, thank you very much, Dr. Kututi, for one class.
Yeah, sir. Yeah, I know, sir. I have also listened to you once. Yeah.
So, first of all, I congratulate you.
Thank you, sir.
For presentation in respect to plant genome editing, because here we are discussing the livestock, fishes as well as plants. So I think one of your slides you presented that you have identified a particular miniature protein, which is similar to the function of Cas9 or Cas12.
Yes.
Whether that is CRISPR-Cas mechanism of function it is following or any other thing.
The mechanism is exactly similar, but it is not a CRISPR-Cas9 system. It's called TNPP, which is an ancestor of Cas12a. It's an ancestor protein. It's a very small protein. So, but mechanism is same. You have to design a guide RNA, and this protein goes and comes.
Mechanism is same.
All same. Yes.
If mechanism is same,
Yes.
You cannot protect IP for this because that IP is there. Not only Cas9 and Cas12, but Cas series of proteins, 4 types of proteins.
Yeah.
And that is the mechanism also. CRISPR, that cluster.
Yeah, but this is not CRISPR actually. This is called ReRNA. This is not CRISPR RNA. This is right end element. The name is different, and we got already patent. It means its patent office has already given and taken consider. But in first examination report, there was no objection on any kind of CRISPR-Cas9 because it does not fall under CRISPR-Cas9 IP protection permission. So we got already that patent. So there is no issue.
Thank you so much. Because you have tried to do something
Yes.
in place of Cas9 and Cas series of proteins.
Yes.
Thank you.
It's not Cas9. Yes.
Okay, thank you, Dr. Kutubuddin. I'll now invite the next presenter, Dr. Periyaswamy Kathiravan, Principal Scientist, ICS National Research Center on Mithun, Matsefima, and former livestock geneticist, IAEA, Vienna.
Thank you to the chairpersons, and I would also like to extend my thanks to the organizers for giving me this opportunity. On this topic of tax to genomics, ICT innovations for accelerated livestock improvement in developing countries. I must admit that I'm not an expert in this field. I have been an end user in facilitating transfer of some of these technologies to some countries in the global south until recently when I was working at the IEE. So I just, from a user perspective, I just tried to give an overview of how these technologies are currently used in different parts of the world and different kinds of production systems. So I was just wondering about the theme of this technical session, like accelerating livestock improvement. Why the livestock improvement needs to be accelerated? So I was just trying to find some reason. So according to the United Nations World Population Prospects 2024, the global human population has reached 8.25 billion by this month, and it is estimated to reach 10.3 billion by 2084, and is expected to stabilize around 10.2 billion by 2100. If you see the population growth here, so it got stabilized in the highest, highly industrialized countries from Europe, North America, and Oceania. But if you see, the growth is still happening in Asia, although it is slightly slow. But in case of Africa, there is a steep increase in the population. And the demand for agricultural products to feed these 2 billion more people is expected to increase by 1.1% per annum. And the demand for livestock products is expected to double by 2050. Coming to the livestock sector, the global asset value is estimated somewhere around 1.64 to 3.3 trillion US dollars, something around 2.57 trillion, similar to that of the crop outputs. And this sector is employing more than 1.3 billion people globally and providing livelihood to around 900 million smallholder farmers, particularly in developing countries, especially in South Asia and Sub-Saharan Africa. So the increasing demand for livestock products is driven by fast-growing economies around in Asia and Africa. So if you see here the global consumption of meat, the past and projected global trend, you can clearly see that the total consumption of meat is increasing slowly in developed countries, but in case of developing economies, it's growing very fast. So both the consumption of meat as well as milk, but still there is a huge difference in the per capita consumption between these two groups of countries. So what does that signify is that the because of the gap, because of the low per capita consumption in the countries that are developing, there is a fast growth of consumption. At the same time, there is a huge increase in the demand. So how this demand is met in these countries, so we can just try to correlate by the livestock population trend. If you see here, the population is increasing at, again, a faster rate in Asia and Africa, while the population is decreasing for most of the important livestock species like cattle, sheep, goat, with the exception of chicken. So the demand is met by rearing more number of animals rather than by increasing the productivity. So that's the point why we need to accelerate the livestock improvement in developing countries particularly. And this slide just shows the gaps in livestock productivity across different parts of the world. So you can see that there is a huge gap in the productivity of milk from the animals that are reared in Africa, Asia, and then North America. So, and similarly, like the situation is almost similar in cattle, sheep, and goat. Probably poultry is a bit of an exception. Again, that reduced difference is mainly due to the industrialization of poultry sector in China and India. So, with this huge difference in productivity between livestock in Asia and Africa and industrialized countries, there is a strong need to accelerate livestock improvement in developing countries to meet the growing demands. So many speakers have already dealt in detail about the challenges of the productivity, challenges regarding the productivity in developing countries, especially the production system is dominated by the smallholders, or either smallholders or by pastoralists, maybe nomadic pastoralists or the transhumance pastoralists, lack of adequate high genetic merit males, the limited access to quality inputs in terms of feed, housing, veterinary care, the unorganized and limited market infrastructure, the tropical heat, high disease burden, and most importantly, lack of reliable data for informed decision making. So, for the last 2 decades, there has been revolution in 2 major areas. One is the genomics, where the cost of sequencing has come down a lot. For example, if we consider the Human Genome Project that started in the early 2000s, the cost of sequencing the human genome was somewhere around 2.57 billion, but now it is possible to have a decent coverage of sequences for about $200 to $300. For example, a 10x coverage. And then it is possible to have the prediction of an animal's genetic merit at an younger age using these genomic selection approaches. With respect to the information communication technologies, these are the diverse set of tools and resources used to create information, store information, process information, and then provide feedback to the end users. So it includes sophisticated systems like the Internet of Things, involving sensors, cloud computing, blockchain technologies, and artificial intelligence. So, which has given an unprecedented opportunity for real-time monitoring of individual animals and also to monitor the herd level parameters. So, coming to the ICT applications in livestock, so it varies. It varies across different kinds of production systems. In the intensive production systems of highly industrialized countries, the systems are very sophisticated, involving automated milking systems, electronic feeders and weighing systems, the IoT sensor-based networks, and then the environmental monitoring. But in case of smallholder and pastoral production system, it's mainly restricted to mobile-based SMS information on different husbandry practices, like husbandry interventions, and then some simplified data recording tools. So in case of the pasture-based systems, like for example in beef ranching, there the applications are still quite useful and interesting. For example, the satellite GPS-based tracking, and then use of drones, and then the low-power wide area network sensors. So as I was mentioning that in the intensive production systems, the automated milking systems, they not only or applied for voluntary milking, but also involves collection of wealth of data, including the milk yield, composition, fat, composition such as fat, protein, and then in some cases like somatic cell count, and also for monitoring the cow behavior and activity. And similarly for the automated systems for measuring the feed intake and body weight, and that can be used for improving the feed efficiency and growth performance. And then the IoT sensor networks, where the wearable sensors like the accelerometers, speedometers, and then several other kinds of sensors help monitoring the physiological and behavioral parameters, and then those information are transmitted wireless for further processing and use in detection of sickness or detecting the heat or stress in the animals. And also the environmental monitoring, like the sensors in barns help monitor the temperature, humidity, and gas concentrations such as ammonia or carbon dioxide, so that can be helpful in implementing the automated control of ventilation and cooling systems to mitigate the heat stress. With respect to this pasture-based production systems like beef ranching, this GPS-based monitoring is also now getting popular. For example, this virtual fencing, where the animals are guided by audio cues. The animals get a neck collar, and then the neck collars, so first of all, the fences or invisible barriers are created using the GPS data, and then when the animals try to